So it's a real pleasure. Thank you, Lynn, for the introduction. It's a real pleasure to be here and to tell you about our research. So today what I'm going to talk about is new techniques that we've developed to try to push the boundary of detection of single molecule all the way towards the animal imaging. And as a follow-up on Stephen's very nice introduction, I just want to remind you how wonderful these single molecule techniques are. And they allow basically improve image resolutions as well as resolving cellular nanostructures in sick cells when they are applied to super-resolution imaging such as PALM or STORM, where, as mentioned by Stephen earlier, you can actually localize individual molecule using mathematical Gaussian functions to improve the overall imaging resolution of the final image. You can also use these techniques in live cells, looking at diffusion dynamic at the nanoscale through the same principle, but this time taking into account the fact that your molecule is diffusing around, and that allows you to detect subpopulation of diffusing molecule, to classify them in different diffusion behaviors, and indeed to access asynchronous and different dynamic molecular behavior at the nanoscale. So these techniques really allow us to peer into cellular process with amazing details at the nanoscale. One limitation, however, of the current techniques and single molecule imaging are that they're really optimized for cell culture tissue slices. And there's always a concern when you work with cells that is related to the fact that your cells and the molecular events that take place on the cultured cells might not truly represent biomolecular events as they occur in vivo. And few examples or few possibilities that, cell line can actually lose tissue-specific function. The fact that usually when you grow a cell on a cover slip, you have generally an absence of extracellular matrix, which might change the way the cell behave, and obviously, generally, cells in culture tend to overexpress tagged biomolecule. So there's really a need to move and to try to move our single molecule imaging from the culture dish all the way towards the animal. And so when you want to do such a move, basically comes with obviously a few complications as well as advantages. So obviously you need to find animal models that are amenable to microscopy if you want to achieve single molecule in live tissues. And you also need to engineer different type of tissues, the aim being trying to reduce the signal to noise, try to improve your signal to noise, and try to bypass the autofluorescence that generally comes when imaging a tissue. And obviously you need to be able to express your molecules or your protein of interest at endogenous level in order for it to represent the normal behavior of the tissue. Now the advantages, so these are all complications that need to be solved. The advantages on the other hand are that you are now capable in principle to work with the native 3D environment of the cells under homeostatic control of the body of the animal, and obviously at endogenous and physiologically relevant expression level, which is key to get a good understanding on the biomolecular process. Now, one overarching advantage of that is if you now work with a live animal, you also have access to disease model, which allow you to look at these molecular processes at different scale and at different representation or different stage of a disease, and you can think about looking at Parkinson, aging, or muscular dystrophy. And for today, what I want to show you is that you can use C. elegans nematodes as an animal model, which are highly amenable to light microscopy, notably because of their easy genetic. And a new technique for single molecule imaging called complementation activated light microscopy, or CALM, which allow a controlled and tissue-specific single molecule detection at physiological expression level by simple knock-in of fusion protein. So I'm going to start by telling you a bit about CALM, this technique called complementation activated light microscopy. This is a technique which is based on the use of a split GFP. A split GFP is basically a traditional GFP which has been split in two parts. The split GFP that we are using has its terminal 11 beta sheet fragment removed, and you end up by removing this fragment with a large fragment, the secondary large fragments of the split GFP right here, which is non-fluorescent. The absence of fluorescence comes from the fact that in the beta sheet that has been removed, there is a key amino acid, glutamic acid 222, which participates to the chromophore formation, and when removed, you end up with the big fragment being non-fluorescent. This split fusion, fluorescent protein has been developed by the group of Jovaldo in Los Alamos, and the very interesting thing is you can actually recomplement the protein and make the GFP active again by simply providing the missing beta sheet fragments as a synthetic peptide in solution. And so, indeed, when you provide this synthetic peptide in solution, it does bind to the split fluorescent protein, and after maturation time, it activates the split fluorescent protein as fluorescent reporter. And the interest in here is you can actually control the level of activation by simply controlling the amount of peptides that you put in solution. So you can titrate how many molecules of GFP you want activated by simply controlling the amount of peptides or synthetic peptide you put in solution. So the way we use this split GFP fusion is basically by taking the big fragments right here and fusing it to protein of interest, transmembrane protein or internal protein, and express them in cells, or as I will show you, express them in transgenic animal. And the principle for single molecule is very similar to classical imaging techniques such as PALM, where this time, rather than using a laser, we rely on the stochastic binding of the synthetic peptides, as well as the concentration and time-dependent complementation kinetic to control the amounts of molecules that we're going to be activating at the cell surface or in cells. Then we can further localize these individual molecules and track them as they diffuse to extract their diffusive behavior, their position, and information about how they behave in cells and animals. So here's an example of an ensemble imaging in cells. In this particular case, we have split GFP fusion fused to a piece of a CD4 receptor, which is transmembrane protein. Different cells are expressing the split GFP fusion. The negative cells here are not expressing. You can verify the expression level of the split GFP by an anti-GFP staining. And when you provide the small peptides in solution at high concentration, you activate all the fusion protein on the surface, leading to the appearance of fluorescent signal, which co-localize very nicely where the protein is. Notice here in these confocal cross-sections that the activation here is purely at the cell membrane. So the peptides themselves do not activate internal split GFP fusion if they're not directly injected in cells. So this is a bulk assay where you use high concentration of peptide, but as I just mentioned, you can actually control the level of activation by actually lowering your peptide concentration. So here's the same cells with a piece of a cell here imaged by bright field, and in this particular case we use total interreflection for imaging. This is the part of the cell area, fluorescence image of part of the cell area before complementation, before adding the peptides, and within 5 minutes of adding the peptide in solution, as the peptide diffuse, binds, and the fluorescent protein mature, you can see individual spots corresponding to individual fluorescent protein, GFP CD4, showing up at the surface, and over time the amount of proteins increases as the maturation takes place. You can, again, using Gaussian functions, go from your raw image where you have the individual point-spread function corresponding to the image of the individual molecules. You can localize them by Gaussian fitting and again do tracking and diffusion analysis. In this particular case, we have an accuracy in imaging in localization precision of about 25 nanometer. On a side note, so this is done with a split green fluorescent protein version. On the side note, we are developing alternative FP variants such as CFP variant and YFP variant. We have had big success in cells for the CFP variant, but the split YFP variants are extremely good for single molecule. And in fact, you can see from this video that complementation takes place at the surface of these cells, and you can indeed easily detect individual fusion protein diffusing at the surface. This is again done by total internal reflections. So using these variants, we think we can actually achieve a multicolor individual molecule through the same complementation activated light microscopy technique. The interest in here is that these different color variants all use the same complementary peptide. So with a single complementary peptide, in principle, you can achieve at least 3 colors, and we're working on red variant on our side. I should mention that all these split FP are actually compatible with photoactivable FP if you want to combine CALM and PALM imaging for multicolor detection. So in addition to cells, we use this split fusion for single molecule imaging in animals. In this particular case, we generally do knock-in of the split fusion to receptors or protein of interest, and we express the fusion through endogenous promoter so that the expression is achieved in particular tissues, and then we generally come up with our synthetic peptide, inject them in the circulatory system of nematodes, C. elegans, and as the peptide diffuses through the nematode, it will reach the tissue and light up the fusion at the specific tissue where the fusion is being expressed. So here's an example of fusion to a CD4 receptor. Again, this is a split GFP fusion. This is a bright field image of the worm, and this is the corresponding confocal image in the GFP channels. You can see in this particular case the CD4 fusion is expressed through a myo3 promoter, so it's expressed in muscle cells. You can see a clear lighting up of the muscle cells upon binding of the peptide. Here's another example which I will develop a little bit later on in the presentation, where the fusion is done through calcium channel, which are expressed on muscle cells again, but also at neuromuscular junctions in C. elegans. Here's an example of a muscle cell where the calcium has been activating after diffusion of the peptides through the worm, again, right at the surface. An interesting comparison is to compare the same calcium channels now through an EGFP, a complete FP fusion. You can see the difference in imaging here. In the case of the split GFP fusion, we light up only the surface channels, while with the GFP fusion, we also get some signal from the channels coming at the surface, intracellular channels trafficking all the way to the surface. And you can see the difference in terms of the image crispness. In this particular case, here we truly only image the membrane versions of the channels and not at all without interference of the fusion coming from inside the cells, coming at the surface. So these are bulk cases, again, where we have high concentration of peptides, but something we wanted to do before moving on to single molecule was to actually show that indeed the activation is specifically due to the binding of the peptide. In this particular case, we take advantage of the fact that the peptide is synthetic, such that it can be modified with additional fluorophore. And when you do so, basically complementation will induce activation of the FP, and as well as positioning of a fluorophore in very close proximity to the fluorescent protein chromophore. And in this case, you can actually use the FP as a FRET donor to the chromophore on surface. So in this particular example, we inject peptides modified with the Alexa 647 fluorophore, and we expect activation at the muscle cells to be visible in the GFP channel, but also in the red channels through FRET. So this is the wide field view of the worm, and when you image both the green and red channel by confocal imaging, you can see indeed that the muscles light up, so that means the peptide has been bound, but you can also see that the peptide diffuses through the worm and has bound to different sort of tissues. So there's a lot of non-specific binding of the peptide, which we never see when the peptide is not labeled, but which is clearly visible here upon labeling of the peptide. And so to get rid of these backgrounds, you can actually use these threads that I mentioned, where you now excite only the GFP, but you look in the threads, you look in the Alexa 647 channels, and you actually, as you can see, and I'll go back to the next slide, you can actually see an effective cleaning out of everything that is non-specifically labeled, and the actual muscles light up very nicely in the Alexa 647 near-infrared channel. And an overlay of the GFP and the red channel, usually gives a nice colocalization. So this, for us, is interesting because it confirmed that you have irreversible binding of the peptides to the speed-GFP fusion in animals, and that we can actually get rid of some of the autofluorescence or some of the fluorescence coming from non-specific binding of the peptide in case you use labeled peptide or fluorescently labeled peptide using this thread detection, basically single per thread, from the GFP to the dye that is now attached in close proximity. So let's go now to the actual single molecule in animals. In this particular case, we use the same approach where rather than injecting high concentration of peptide, we actually lower the concentration significantly so that we can achieve individual molecule detection. In this particular case, we use a technique called HILO, or highly inclined and laminated optical microscopy imaging, which is a light sheet technique that allow us to, in fact, image muscle cells right at the interface between the muscle cells and the basal lamina within the worm. And as you can see now from this video, when you lower the concentration of peptides to sufficiently low amount, you can actually detect individual molecules. So the little white spot that you see on the left-hand sides are basically individual channels at the surface of the muscle cells lighting up, and they can be tracked. On the right-hand side, you can see the tracking path. The image is a bit lower resolution, but you'll see on the next slides corresponding to tracks of these particular channels at the muscle cell. So we can actually indeed now do tracking. So here's a representative example of different tracks at the surface of a muscle cells. These are example of the signal-to-noise ratio that we have in live animal at the surface of the muscle. Usually we use a probability distribution of the square displacement analysis to identify subpopulation of different protein diffusing. Here are some of the tracks. And the actual localization precision, I forgot to mention that, is about 30 nanometer in the live worm. Our statistics usually are performed on a really large amount of trajectory on the tenth of thousands and in multiple worms, and we can in fact deduce different kind of diffusion mobility in this particular model receptor, which is again this CD4 fusion to speed GFP. We could define basically three types of subdiffusive behavior, 10% Brownians, and the rest of them being slow diffusing in confined domain. So these were model system. What we were more interested in was to apply this to actually disease models, and that's why we focus on this calcium channel, looking at the diffusion at the muscle surface, as well as the neuromuscular junctions of C. elegans model for muscular dystrophy. In this particular case, we take our speed GFP and we fuse it to a piece of the calcium channel called the alpha-2 delta subunit. And similarly, so this is a knock-in fusion, and we replace the endogenous calcium channels with this fusion in worms, and we basically have two types of imaging conditions where we immobilize the worm on beads and where we image the worm between contraction. That's what we call a resting state, and you will see on the right-hand side of this movie a secondary way of imaging the worm, which is through the use of an anesthetic where the muscles is induced to contract. And as you can see again from the left-hand side, the little bright spot that you're seeing corresponds to individual calcium channels being activated at the surface under what we call the resting state. If actually the movie plays again, you'll notice that the the worm is moving a little, so we actually do imaging in between contractions. And then in the case of the drug treatments on the right-hand side, we have the worm being anesthetized, so there's little movement. So you can see here the arrow pointing out at some movement, so we usually track in between these contractions. So from these movies, we can now extract diffusion coefficients and information about the trajectory of these different calcium channels. Here I should mention, before I move to the next slides, the fact that we have typical single molecule features such as blinking, which basically confirm that indeed we're looking at individual channels in the live worm. So here are a few examples of trajectories that we obtain. And we can build statistics to analyze the diffusion coefficient of these different channels. In general, we find two population of channel: slow diffusing channels as well as fast diffusing channel. The slow diffusing channels tend to be confined into 80 nanometers nanodomain, so you can see a clear confinement effect when you do the tracking. And the rest of the populations, which is about 10% of the channels, tend to diffuse freely over short distance around 230 nanometers. So that's and I'll go back a little bit more into details in the next slides, in a few slides, about how we can now use this diffusion coefficient to compare the behavior of the channels in a normal worm and a dystrophic muscle. But before I move to that, I want to show you that you can also use column imaging as well as dual color to localize more precisely where your molecule is diffusing in a live animal. In this particular case, the calcium channels that we've tagged with this PGFP are expressed in the muscles, but they're also expressed at the synapse, at the neuromuscular synapse in the worm. So the worms have these long nerve cords along the body, where muscle cells actually send projections and make synapse to this nerve cord. And our calcium channels is expressed at the muscle surface here, but is also expressed to some extent at the neuromuscular junctions. And to identify the channels that are at muscular junction, we use dual color imaging through a 2-cam system with 2 EMCCDs. In this particular case, the synapses themselves are labeled through an acetylcholine receptor fused to tag RFP. And you can see that these tag RFP lineups in a line corresponding to these nerve cords, and the synapses themselves are labeled in these red spots. And at the same time as we image the synapse, we can also image in secondary channels the GFP fusion activated by complementation by CALM. And you can overlay the trajectories of the channels with the position of the synapses to identify which channels is actually diffusing at the synapse. And you can do this for excitatory synapse, inhibitory synapse, and so on. So in this case, again, this is now helo imaging with dual color, 2 lasers, and we split the signal through an AND or 2-cam system onto EMCCD camera, and this is imaged simultaneously. So to go back on a little bit more details on the diffusion properties of these channels in normal and dystrophic muscles or contracted muscles, you can see that, as I just mentioned before, these channels tend to be confined. 80% of the channels tend to be confined in 80 nanometers nanodomain. The fast channels themselves diffuse much faster. And then when you compare now this diffusion between resting muscles and contracted muscles, we actually observe a very dramatic decrease in the diffusion properties of the slow population, where the channel seems to be pinned down or immobilized, if you want, by the muscle contraction. And we interpret this, if you want, as a feedback mechanism that is linked to localizing the flux of calcium as the muscle contracts in order to try to potentiate the muscle contraction by enhancing interaction with other kind of calcium channels, other kind of calcium channels that are calcium dependent and that are localized just below the surface of the muscle cells. We also observe a reduced fluidity during contraction through the decrease in diffusion coefficients of these fast channels that you can observe here. which we believe is related to the increased stiffness of the muscle membrane during contraction. And now, to make a long story short, what we observe when we remove or when we analyze the same channels in dystrophic worm, where the key protein called dystrophin, which is basically an organization of protein and protein complex just below the membrane that link the cytoskeleton of the muscle cells to the extracellular matrix. When this is being removed, we see completely different behavior of these calcium channels, which rather than becoming slow during contractions, keep diffusing into the nanodomain, indicating that there is probably lack of feedback regulation of the calcium properties by the absence of dystrophin. And then to finish, I'd like just to show you that you can, as Stephen presented earlier, you can actually use all the tricks in the book from single molecule to actually quantify the number of protein per domains or by single molecule counting. So here this is using high concentration of peptides to activate as much as possible of calcium channels at the surface of muscles, and basically going down and letting the molecule bleach and counting the number of bleaching steps corresponding to one particular type of nanodomain. So we sit, if you want, on one type of nanodomain right here and let it bleach up to the point where we reach individual molecule and then count the number of bleaching steps, which allow us to actually evaluate that we have at least 10 calcium channels per nanodomain for the slow diffusing one. We can also use this single molecule localization to do spatial distribution analysis or near neighbors distance analysis, try to evaluate how are the calcium channels at the muscles of live animals or live C. elegans are being distributed. In this particular case, we found that this distribution of the nanodomains themselves is not random, but is actually more dispersed than random, suggesting that there is a very well organized, very good organization of the nanodomain at the surface of the muscles. And so I like to finish basically by again summarizing the fact that you can use this split GFP and COM macroscopy to target and image individual molecule in cells, as well as in animal. You can achieve multicolor imaging through the developments of split GFP or split FP variants, but also through FRET processes, or by actually combining with traditional imaging of other FPs. And ideally and potentially this particular technique applied to live animal really allows now biologists to explore the basic principles of homeostatic control as well as the molecular basis of some disease at the nanoscale by allowing imaging in live animal. And with this, I will take questions. This actually the group in my lab which are working on this particular project, and this is a very nice collaboration we have with Jean-Louis Bressreau and Hong Zhang at the University Claude Bernard in Lyon. These are the funding agencies, and I'm happy to take questions at the end of the webinar.
Thank you very much, Dr. Pinot, for your presentation. It is my pleasure